So coming to the disease itself, so non-alcoholic fatty liver disease is basically defined as accumulation of fat in the liver of individuals who have a history of no or moderate drinking, alcohol drinking. Although this definition is now being challenged and a new concept is coming for this. Anyway, we'll start with the old classical definition of NAFLD. So this is how NAFLD starts. So it starts from accumulation of fat in the liver, which is the benign state. So it's not a clinically serious condition. The fat exercise and diet regulation actually reverse a lot of fatty liver problems. The problem actually when it is clinically relevant is when this progress to a spectrum of this disease known as NASH, non-alcoholic steatohepatitis. The problem with this phenotype is because in this NASH condition, there is an increased death of hepatocytes, there's increased inflammation, the ALT levels rise, and then you have a more risk of developing fibrosis and cirrhosis, and ultimately it can lead to hepatocellular carcinoma and liver failure. So whatever the drugs are being developed nowadays are basically focused on either stopping the progression of benign steatosis to NASH or trying to reverse NASH back to a benign steatosis state or normal liver state without progressing ahead to the cirrhotic state. So NASH is the main focus of drug design nowadays. So NAFLD itself is a very multi-system disease. A multi-system because its origin is also diverse. It can be because of different environmental factors. It may be because of metabolic disturbances like insulin resistance, visceral adiposity, endocrine dysfunction, which I'll be talking in my slides also, which is a major contributor for NAFLD. And of course, gut dysbiosis, and to some extent, genetic causes have also been attributed to the development of NAFLD. So it's both environment and genetics, although environment plays a much more bigger role in development of NAFLD. But after NAFLD is developed, it itself has a lot of effect on other diseases. So it's a problem not only because of virtue of being a chronic liver disease, but it also increases the risk of developing diabetes, CVDs, and hepatocellular carcinomas, osteoporosis. So it's linked to a lot of diseases, metabolic as well as diseases of cancerous origin in humans. So its complication, its scope of effect and medical condition actually increase a lot. So if we talk about the timeline, so how, and this also explains you why, although it's a disease focused on the liver, but the endocrinologists are very interested in disease. So if you see the origin of NAFLD itself, it was actually the first demonstration of this fatty liver changes in the in patients who are actually described by Addison, who actually is known for the discovery of Addison disease. So he showed that he was the first in reporting alcohol-induced liver histology changes, and that was development of fatty liver in alcohol-induced liver diseases. Then other researchers also showed that development of fatty liver is the first step towards cirrhosis, either because of alcohol drinking or due to diabetes. So it's a common factor. Hepatic steatosis is a common factor in both alcoholic liver disease as well as non-alcoholic liver disease. Then in 1980 was this landmark paper by Ludwig, which actually showed and demonstrated the NASH itself and showed that this is a type of cryptogenic cirrhosis, which is very much correlated with patients who have type 2 diabetes and thyroid dysfunction. So this was a seminal paper in this field. And then later, Leonardo actually showed the bright liver syndrome thing, in which we really use this ultrasound to show the fatty livers, signs of fatty liver by this bright syndrome, in which echo pattern, it shines bright white in the ultrasonography. And finally, now the whole concept of NAFLD is being changed. And there was a consensus statement by Sanyal et al. And they actually showed that it should be renamed, the disease should be renamed as NAFLD. The reason is that there were two reasons for that. They actually advocated that NAFLD itself is a disease defined by exclusion criteria. It's people who don't drink alcohol and develop fatty liver are called NAFLD. It's not something which the disease has been included in this disease. And of course, it's a stigma in Middle Eastern country of using the word non-alcoholic. And finally, most of the non-alcoholic fatty liver disease patients do have some symptoms of metabolic syndrome. Either it may be type 2 diabetes, CVD, adiposity. There are minor population of NAFLD and NASH who do not have metabolic syndrome, and these are called lean NASH patients. But a majority, 80 to 90% people do have, especially, and this is actually biased towards the Western scenario in which most of them do have metabolic syndrome. So they have now advocated to change the term as MAFLD, so metabolic dysfunction associated fatty liver disease. And of course, NASH to be changed to MASH, which is the same thing, metabolic dysfunction associated steatohepatitis. So if you see the increase in the study of NAFLD, I'll still use the old terminology so as to do not confuse with MAFLD and NAFLD, but it's actually the same thing. So if you see the increase in the research in this field of NAFLD, it has grown tremendously since Ludwig described NASH in 1980. If you see the last 10 to 15 years, there is a huge increase in the research of this disease. The reason is because this is becoming a major health concern all around the world, in both Western world and in the Asian continent itself. And because I have shown earlier, it's a risk factor for type 2 diabetes, which is a major problem. So a lot of research is being now directed how to control NAFLD. So if you see the latest global trend of prevalence of NAFLD, you'll be surprised to see that more than 30% of world population is presumably having NAFLD. And initially it was thought that it is basically a disease concerned with the West, in which there's a lot of calorie intake and stuff, and it's basically disease of the developed world, but it's not the case now. If you see in Asian continent, in South Asia, which is our Indian subcontinent, including Pakistan, India, and Bangladesh, actually it is the prevalence percentage is actually more than the global average. So it's a big problem in our subcontinent. And the highest prevalence is in Latin America, which is around 44.4%. So everywhere it is increasing, and it is a major health problem, especially in the Asian countries and South Asia, Indian subcontinent per se. So, so this was the prevalence of NAFLD. What about NASH, which is a clinically relevant spectrum of NAFLD? So in South, if you see, it is global prevalence of NASH is 5.27%. And in South Asia, in our Indian subcontinent, it is 5.42. So it's again more than the global average. So it's a big problem here. And among the NAFLD patient, the prevalence of NASH is around 16%. So this is the latest statistics which was published in Hepatology this year. And as the disease itself is exponentially increasing, so is the valuation of the drug market of NAFLD. So still we don't have any drug, but a lot of big pharmas are actually moving ahead to find new drugs. The reason is the evaluation of the drug market. So if you see in what has been predicted in 2021 to 2030, it has increased around 4.8%. So it is now presumed that the drug market valuation of NAFLD drugs will be around 28 billion USD. So it's a big drug market for the pharma companies, and because it's a big problem all around the world. So unfortunately, still we don't have any FDA-approved drug for NAFLD or NASH, but there are some preventive measures which do help in mitigating the progress of this disease. And some of these are basically dietary changes, weight loss, bariatric surgery, modulating gut microbiota, nutraceuticals, proper sleep, and physical activities. So they do actually work quite nicely. As far as benign steatosis is concerned, they are quite good. But in reversing NASH, they have limited efficacy. And as the NASH progress, you actually don't get that much benefit from exercise and dietary changes. And so you need some drugs target to identify. And also the compliance is also an issue for most of these stuff here as a preventive measure. So with this in mind, there is a urgent need to identify pharmacological drugs and drug targets to treat NASH and NAFLD. So in a targeted pharmacology approach, there are two ways in which you aim to design a drug for clinical use. So first way is the traditional hypothesis-driven approach, in which you have observation which you make on the bedside, or if you're a researcher, you make observation by studying in animals and going through the literature. And then you validate some of your hypothesis target, which you hypothesize maybe playing role in the disease by using either human cell lines or animal small animal experiments, preclinical models. And if you get the hit right, then you go for the clinical trial. So this is the traditional hypothesis-driven research. But with the boom of the omics technology, whether it be metabolomics, transcriptomics, genomics, so these omics technology have the power for unbiased screening of drug targets. So what you do have is now you do a microarray. Anybody in the world who has done a microarray or a RNA-seq in a population of population of in humans who have NAFLD or NASH, and you take these microarray data, you analyze that what are the genes which are being upregulated or are being downregulated, and then based on that you make a hypothesis that possibly these genes may be playing some role in the pathogenesis of the disease, in this case NASH. And then you do a preclinical trial with animals and cell line models, and you come up with a number of drug targets which can use for targeted clinical trials. So this is the new way in which you can actually reduce time, and actually you can get a lot of target in one go. And the beauty is that you take human set as the starting point. So you don't have to worry that your initial experiment is in animals. You take a microarray data set from human population of a disease, and then you validate in animals, see if it is conserved across species, and then you again go for the clinical trial. So this is a much more smarter way to find. But of course, hypothesis-driven research still is going on, and that is a very rational way of going in most of the lab for drug design. So our lab approach for target identification in NAFLD involves both these processes. So we use both hypothesis-driven approach as well as the big data-driven or the omics-driven approach. So we have nailed down few leads which we have derived from the hypothesis-driven research, and these are basically on hormone-based therapy for NAFLD and targeting extracellular nucleic acid, for which I'll describe in the coming slide. And we have also used human microarray transcriptomics data set of NAFLD and NASH patients to come up with some new targets which can be used for mitigating the progress of NASH. So I'll first go through the hypothesis-driven approach which we took, and in that category, I'll go to the hormone-based therapy which we are working in. So this hormone, which is actually a very dear hormone to me because my PhD was also on thyroid hormone and brain development. So when this story of my working as a person working on NAFLD started when I was doing my postdoctoral work at Duke Medical University in Singapore. And when I was doing my postdoc, I came across a lot of articles in which they showed that patients, hyperthyroid patients, have a more susceptibility to develop NAFLD. So there are a number of papers were coming. So that actually ignited my imagination to see whether thyroid hormone has a direct role in regulating liver fat. So at that time, we all knew that thyroid hormone is a inducer of basal metabolic rate. It increases energy expenditure. But there was nothing known if thyroid hormone itself can regulate lipid turnover inside the liver cells. So this was the question I wanted to address. So what we do is that we generally take, for the initial starting point, we take human hepatic cells, load them with fatty acid to mimic a NAFLD condition, and then we add thyroid hormone to see how these hormones act to decrease the amount of lipid inside the cells. And we also use animal models in which we give them high-fat diet or high-carbo diet and see if thyroid hormone can reduce the hepatic steatosis and NASH progression in these animals. So the discovery which I made during my postdoctoral work and which became the rationale of leading up to all this work which I am doing today is that we showed that thyroid hormone indeed induce a very selective cellular process known as lipophagy. So till before that, it was not known whether thyroid hormone can increase lipolysis in the liver. And in adipose tissue, the only thing which was known is that there were lipases, which were hormone sensitive lipases, which increases liver lipid turnover in the adipose tissue. So what we were able to show that there is a process known as autophagy. Autophagy is a cellular process in which cellular organelles known as autophagosome come near the proximity of lipid droplets or the triacylglycerol lipid droplets, and they chew them. So what they do is that the membrane fuses, they take lipid one by one, they engulf the lipid, and then they fuse with the lysosome. So this is the process actually which happens. So these are double membrane structures which engulf the lipid droplet and fuse with the lysosome, release free fatty acid, which can now go into the mitochondria for beta oxidation. And that is how you get rid of the liver fat by thyroid hormone. So this process, because it was the autophagy directed against the lipid droplet, it was termed as lipophagy. And what this EM graph shows you is that this is a control mouse liver EM, and this is a T3 treated livers, T3 treated mouse liver. And these black structures, if you magnify them, these are basically autolysosomes. This is the autolysosome containing a triacylglycerol lipid droplet inside it. And it is also, you can see a double membrane structure strong. This is the autophagosome fused with an lysosome structure. And if you see this one here is basically a lipid droplet and this myelin type structure, this autophagosome structure going inside this lipid droplet. So basically it fuses with the lipid droplet and it chews it and bring it to the lysosome for degradation. And that is how the beauty of cell biology comes into the picture, and how you can modulate the cellular process so as to decrease the lipid load inside the liver. And this is the first step to stop the progression of benign steatosis to NASH. So this was in a normal physiological condition. So we tested it in a preclinical NAFLD model. And this is basically, these are C57 black mice, which have been fed high fat, high carbohydrate diet. So what happens that when you feed them high fat diet for 16 weeks, they develop all the features of metabolic syndrome. Most of the features, they have adiposity, they develop type 2 diabetes, and definitely they have fatty liver. So what I'm showing here is that when this is a normal chow diet fed mouse, this is a high fat fed mice, and you can see the lipid droplet, the liver TG level goes up. But when you add T3, you inject T3 in these mice, you can bring down the lipid droplet content in the lipid content in the liver. At the same time, if you remember, I have shown you that I've showed you in the previous slide that whenever there is an autophagy, you degrade lipid, the lipid goes into the mitochondria for beta oxidation. So the mitochondrial activity increases. So what this graph is showing you actually is the oxygen consumption rate. So if you isolate the hepatocyte from these animals, we were able to show that T3 can increase the oxygen consumption rate of mitochondria in a time-dependent manner. So they were all correlated, the decrease in lipid content in the liver and increase in mitochondrial activity. And this was all related to the autophagy induction by T3. Now, fortunately, after doing these experiments, there was time to go for the translational aspect. So the question was, does thyroid hormone decreases liver fat in human, in a patient with NAFLD? So we did a phase 2B clinical trial. I was a part of this. And what we did was we gave low dose levothyroxine in patients with type 2 diabetes and NAFLD. And although the effect was modest, so it was a 2% reduction in liver fat, because the problem is we cannot give high doses of T4 because of the adverse effect on heart. So the amount of T4 given was titrated so that the TSH level was between 0.3 to 1.7. And so they were not hyperthyroid at itself, but near the lower range of the TSH. And we actually were able to show that thyroid hormone does decrease liver fat, and of course, it also increases insulin sensitivity correlated with it in humans. So this was the first demonstration which started from a preclinical trial with animals. We translated it into humans, and then it is really gratifying to see that now the pharma companies have taken up this concept of using thyroid hormone for NAFLD and NASH, and thyromimetics are poised to be the first FDA-approved drugs which may soon be in the market, because the company, this is Metrigel, they have designed a thyromimetics. So we all know we cannot give thyroid hormone to patients because of the thyrotoxicity. So what the companies have done is that they take the concept from the work we have done on thyroid hormone, they modify thyroid hormone molecule itself in a way that it becomes specific to a receptor, thyroid hormone receptor, which is only expressed in the liver. So you don't have side effects in the heart and the skeletal muscle. So these are liver specific thyroid hormone mimetics, and they are a liver specific thyroid hormone receptor mimetics. So it only binds to thyroid hormone receptor which is expressed in the liver, not in the heart and the skeletal muscle. And then you can use the pharmacological doses of these thyromimetics, and they have really cleared very well in the phase 3 clinical trials, and they have been granted what is called the priority review in the FDA. So most probably it will take another 8 months for the FDA to approve them as the first drug for NAFLD and NASH. So it's really gratifying that work we started in animals is really made it to the drug market now. So, and this news is as recent as September 13th. So just a month before it was granted this accelerated review by FDA. And other than thyroid hormone receptor mimetics, good results are coming from other nuclear receptor ligands like FXR and PPR. But some of thyroid hormone receptor are possibly will be the first to make it to the market. So what we are doing now, based on this, we are using this thyromimetics. So the thyromimetics resveratrol, as I've shown you, is a very specific thyroid hormone analog which target only the liver resident receptor for thyroid hormone. So we all know that NASH progresses to hepatocellular carcinoma, and it may go via cirrhosis or can directly turn into a liver cancer. So what is not known is whether thyromimetics or thyroid hormone can prevent the progress of NASH to hepatocellular carcinoma, and that is what my lab is currently working on. So it's still initial work. So probably next time when we meet, I can update you on the work, on the data we get from this. And but other than that, so this was the first part which I covered, and the next hormone which we are working, and we do have some data on this, is DHEA. So DHEA is basically dehydroepiandrosterone, is basically a sex hormone precursor, which is basically converts to estrogen and testosterone. And this hormone has been, for many years, it has been thought of as a very wonderful hormone which has all health promoting effects. It increases lifespan, it improves insulin resistance, and treats depression. So there were some studies, epidemiological studies, which showed that in people living in some islands like Okinawa Islands and some islands in which they generally the population is centurions, they generally cross 100 years, their DHEA levels was much higher than the people living in another part of the world. So they have definitely were shown to have some effect on increasing lifespan. And weight loss and insulin resistance, improvement in insulin resistance was also shown in many studies in humans as well as in animal models. So what we were amazed to see is that these levels of these hormones actually were shown to go down as NASH progresses towards fibrosis. So somehow these hormones, DHEA basically looks like it may have an anti-fibrotic role in the liver. So because the first study in hepatology in 2008 and then recently in gut 2019, they show as the liver fibrosis progress, the level of these hormones go down. So we ask, what if we give this hormone as a supplement in an animal model of NASH? Can we reverse or can we mitigate the progression of NASH? So this is the question we ask. So what we made is this NASH model we make is we take mouse strain, which is C57 black 6. So we give them normal chow diet, or we give them this NASH inducing diet, which is called high fat methionine choline deficient diet. So the concept is, when you don't have methionine choline in the diet, you cannot export VLDL from the liver. So, and over that, when you're giving high fat, the liver will start accumulating lipid, and it may not, and it will not be able to secrete it out. So it slowly it will develop NAFLD, and within 6 weeks of feeding, you will have a full-blown NASH develop in these animal models. So these are very good models to study advanced NASH. So you'll have fibrosis also within 6 weeks. And if you keep them for longer, you can even see signs of cirrhosis. And some people have also shown spontaneous occurrence of SCC in these models. So you see, so what you can see is that the serum DHEA, so DHEA actually is sulfated in the body. So it exists both in the DHEA free form and the sulfated form. So you see in the normal chow diet, you have DHEA, and then it goes down if you feed the animals high fat diet with MCD. So it mimics the human condition. In humans also, in NASH, the DHEA level goes down. In animals also, feeding them a high fat methionine choline deficient diet, the DHEA level goes down. And when we give them DHEA exogenously, it is restored back. And what you can see is the very good correlation with ALT level, serum ALT level. So you see, as in this NASH model, the ALT level shoots up, and it can be brought down when you give DHEA in these animals. So DHEA definitely has a liver protective role in these animal models of NASH. And when and then, because fibrosis was the thing which was correlated with DHEA level, we wanted to see how it affected the NASH-induced fibrosis, administration of DHEA. So these genes, COL1A1, COL1A2, COL13A1, are basically genes which are induced in the liver when the fibrosis starts. So you can see that when you have a HFMCD diet feeding, which is this black bar, these expression of all these genes go up, and this can be mitigated when you give DHEA to these animals. And the hydroxychloroquine levels, which is basically tells you how much collagen deposition is occurring in the liver, is also showing a beneficial trend with DHEA administration. And of course, these markers are also a fibrosis marker, and all of them showed a similar trend that the fibrosis marker go up with these diet, and they can be brought down when you give DHEA. And of course, this is a picocyrus red, which shows you the deposition of collagen and fibrosis. So you have increased fibrosis in these animal models of NASH, and this could be mitigated in the animal NASH model mice which were administered DHEA. So in summary, what these results show is that endocrine dysfunction is intricately linked with NAFLD progression, and hormone replacement and hormone mimetics hold a curative advantage in human NAFLD NASH treatment. So it could be hypothyroidism, PCOS, growth hormone deficiency, and of course, adrenal deficiency, DHEA deficiency, which can correlated with SCC and NASH progression. Now, I want to switch gear and show you another aspect of hypothesis-driven research in which we are targeting inflammation as to control NASH progression. So we all know that in NASH, there is an inflammation, and there is death of hepatocytes. So whenever the hepatocyte die, they release substances or molecules which are called damage-associated molecular patterns. So these can be the RNA from the dying cells, mitochondrial DNA, or other molecules, which are basically, what they do is that they come out of the injured cell and they bind to a specific receptor on the adjacent cell and increase the nuclear translocation of this protein known as NF-kappa B. So NF-kappa B is basically a master regulator of inflammatory response. It binds to the promoter of a number of cytokines and chemokines and increase the inflammation in the tissue. So the idea is, can we block the release of these DAMP from an injured cell and stop the amplification cycle of inflammation? So one of the DAMP we are focusing is extracellular RNA, which are released from the injured cells. So there were a lot of research which showed that in myocardial infarct, extracellular RNA are the major driver of cardiac inflammation. But it was not known whether the same process also occur in NASH. So what we did is that we use enzyme known as RNase A. So we all know that RNase A is the enzyme which degrades RNA. So the concept is, if we give RNase A to the animals, can we degrade the extracellular RNA and thereby stop the binding of this eRNA to the receptors and decrease the inflammation process. So this is the human hepatic cells which we started with, and you can see if we add palmitic acid, palmitic is saturated fat, which is basically related to the lipotoxicity we see in NASH. So when we add palmitic acid to the cell, so this is the vehicle cell, this is a palmitic acid treated cell, hepatocyte, and this is a palmitic acid with RNase 1 treatment. So RNase 1, what you see is that cell viability decreased with palmitic acid. Saturated fat causes lipotoxicity, and this could be basically attenuated by adding RNase 1. And if you see the abundance of extracellular RNA, whenever you add palmitic acid, the extracellular RNA in the conditioned media of the cell increases. So there is a direct correlation with the amount of extracellular RNA you have outside the cell and the viability of the cell inside the culture media. Next, what we did was to see whether the same thing also has a correlation with the NF-kappa B translocation. So this is a confocal image which shows you that in the vehicle-treated cell, NF-kappa B is basically in the cytosol. So you see all these holes here are basically nucleus where the protein has not translocated. But whenever you add saturated fats in these hepatic cells, this protein, pro-inflammatory protein, goes inside the nucleus. So when you overlay this red, which is the nuclear stain, and the NF-kappa B stain, you get yellow stains here. So basically, all NF-kappa B is now inside the nucleus. But whenever you use RNase A1, you actually stop that process. So what RNase 1 is doing is basically stopping the extracellular RNA release, and also its action on its receptor to increase NF-kappa B translocation and the expression of pro-inflammatory cytokines like TNF-alpha and IL-6. So that is how RNase 1 is working. And finally, this was cell line. We replicated it in animal models, and the same thing happens when we add RNase 1 in animal fed with HFMCD diet, which is a NASH-inducing diet. We can reduce liver TG levels, reduce ALT levels, reduce the expression of TNF-alpha, which is a cytokine associated with NASH progression, and also the NAFLD activity score was decreased. So in summary, extracellular RNA we showed serve as a damp and aggravate lipotoxicity-induced liver injury in case of NASH, and extracellular degradation, extracellular RNA degradation either by RNase 1 or by using specific inhibitors of the receptor which bind eRNA, which in this case are known as toll-like receptor 3, may act as a novel treatment modality for NASH. And so we are working more on this aspect now. Now, the last part is basically using big data or omics driven target identification for NAFLD and NASH. So the concept is simple. We take human public data set which are publicly available. So you don't have to pay anything. They are on the websites hosted by NCBI. And then you just do bioinformatic analysis, find out which were the pathways in human NASH are upregulated or genes which are upregulated, downregulated. Then what you do is that you validate these gene target in animal model. So you see, is there species conservation? Because if it's conserved in humans and in mouse, definitely they are something relevant and you can target them. And then finally use functional validation. If it's a druggable target, you use drug. If it's a genetic target in which we don't have drug, we use knockdowns or knockout mouse. And then you can come up with a target which has been validated both in human microdata sets and in animals, and can go further for clinical trials. So we using this omics based technique, we identify two targets. One is AKR1B10, which is basically L2 keto reductase, and CFTR, which is cystic fibrosis transmembrane receptor, which was shown to be significantly upregulated in human NASH. So the thing is, they are upregulated in NASH, but not benign steatosis. And that is the turning point in which we are basically interested. So what happens specifically in NASH that becomes a clinically serious condition? So whatever genes are upregulated in NASH are actually something which we have to target. So you can see here, This is these are control patients. These are NASH, patient with NASH, and these are benign cirrhosis. And they are both AKR1B10 here. And if you see CFTR, they both are upregulated, significantly upregulated in human livers. So we collaborate with bioinformatics colleagues in Duke University Medical School in Singapore, and they help us to do a bioinformatic analysis and give us the main targets which are upregulated or downregulated so that we can work on this. So this is the data analysis which they use is basically PCA component analysis, sample outlier removal, the adjustment for age and sex, and finally they use the ANOVA platform to come up with a set of genes and pathways which are up or down regulated in human NASH and NAFLD condition. So after we come up with these two targets, what this is just give you a brief introduction what these genes are. So AKR1B10 basically is a human protein coded by AKR1B10 gene, which converts retinol into retinol. So when this gene go up, you have more retinol in your serum, less of retinol, which can be converted into retinoic acid and activate the retinoic acid receptor. So this has something to do with vitamin A metabolism. So vitamin A and if you see the clinical literature, vitamin A metabolism indeed is affected in NAFLD. So you can see the beauty how this technique of finding genes actually correlate well with the clinical condition. And then the other, this was a little surprising gene, this was CFTR. So we never thought CFTR may have implication in NASH as such, but it came as a top upregulated gene in NASH. And we showed that actually inhibiting CFTR helps in mitigating NASH, at least in the preclinical models. So what I'm showing you here is basically the validation of these gene in animal model of NASH. So this is basically the same HFMCD diet model. So here we use two models, animal models. So this is a HFMCD model of NASH in which you can see with increasing time, the AKR1B10 expression. So AKR1B10 in mouse has a ortholog known as AKR1B8 also. So there are two genes. In humans, we have one. In mouse, we have two genes which actually work the same way. So both have the same catalytic component. So both of these two genes actually expression goes up, which mimics nicely what we see in humans. And same thing happens with CFTR also. So CFTR also goes up by 6 weeks in HFMCD model of NASH. Now the other model we take is a Western diet model. So this HFMCD model is actually mimic more lean NASH, and the Western diet model mimic more of the metabolic associated NASH. So these models, Western diet models, do develop obesity and type 2 diabetes also. And this model also you can see AKR1B8 levels going up, and CFTR levels also takes time in these models because they take around 24-28 weeks to develop NASH. Unlike HFMCD model, which can develop within 6 weeks. So both these genes were upregulated in two models, animal models of NASH. Now, the question is, what we inhibit these genes? Can we mitigate NASH? So is there any functional relevance of these genes? So we use an inhibitor of AKR1B10. This is actually a curcumin analog. This is called BDMC. It's a curcumin analog, which is an active ingredient of haldi. So it's a active ingredient of haldi. And so when we use this active component BDMC and this has been shown as an inhibitor of AKR1B10 in vitro studies. It actively binds to the AKR1B10 and inhibits its activity. So this is a HE staining, and you can see increased lipid in the HFMCD mouse liver, but the lipid droplet content goes down if we give them BDMC. And this is a picocyrus staining, red staining, showing you that fibrosis also goes down when you give this AKR1B10 inhibitor BDMC. And the same thing for inflammation marker F480, which is basically tells you how much macrophage infiltration is there in the liver. And these are the this is the quantitation of picodust staining, and this is actually a protein blot of showing the expression of cytokines in the liver upon induction of NASH and when you use an inhibitor of AKR1B10. So you can see that as whenever you give a BDMC, which is an inhibitor of AKN-B10, you can actually mitigate the increase in cytokine expression. So now this was a drug-based inhibition. We wanted to confirm it with a genetic knockdown study so that we are sure that it's not an off-target effect of any drug. So what we do is that we take human hepatic cells and we administer siRNA against AKN-B8 and B10. These are mouse hepatic cells. And when we transform these cells with transfect these cells with the siRNA, you can see the expression of cytokine goes down whenever. So this is a vehicle column, and these are the knockdown columns. So AKN-B8 knockdown, AKN-B10 knockdown. So genetic silencing of these two genes also helps in attenuating the basal expression of cytokines. And the same thing we found with CFTR inhibitors. So CFTR inhibitors are available for research purposes. And what I'm showing you is that this administration of CFTR inhibitor in a HFMCD model actually decreases liver fat content significantly, and also serum ALT levels. And these are the photos of the liver, and you can see that the fatty appearance really went down with the CFTR inhibition in HFMCD. Now, CFTR, we don't know, is it the hepatocyte-specific CFTR? Because CFTR actually is expressed in cholangiocytes in the liver. It's not expressed in the hepatocyte. Although there have been some report that in NASH condition, there may be expression of CFTR in hepatocyte. But classically, CFTR is a gene which is expressed in cholangiocyte. And very little is known about the role of cholangiocyte or the cells which line up the bile duct in NASH progression. So it is quite interesting, and we are pursuing the role of cholangiocyte based on the results we're getting with CFTR. So what we are getting is that whenever we add CFTR, there is a decrease in the chemokine expression. There is a decrease in the fibrosis genes which are being induced in NASH, and also F480 staining and Cyrus-Red staining. So definitely CFTR is doing something in NASH, and inhibiting its action is basically helping us to stop the progression of NASH. But we don't know what are the cells exactly whose CFTR is doing this. Is it the cholangiocyte specific CFTR or is the hepatocyte specific CFTR? And that is something which we are basically working on. And that also ignites the concept of many of the cells which have been unappreciated in NASH, and cholangiocytes are one of them. So there is something with the bile duct cells which really is affecting the NASH progression. So at last, the summary is for this part is that omics-based discovery platforms serve as a high throughput, unbiased way to discover novel target for NASH and treatment. Upregulation of AKN-B10 NASH suggests a perturbed retinoic acid signaling, and identification of CFTR in NASH highlights the importance of cholangiocyte in NASH progression. And at the last, I want to acknowledge all my lab members who have worked on these projects. And the funding agencies, Wellcome Trust, ICMR Sub, and our collaborator at Duke University Medical School, Singapore. Thank you very much.